Hello, my name is Isabella de la Paz, and today I will be presenting based on Krimmel and Pierce's scientific paper, which was published in 2013, Sticky Plants Trap Insects to Enhance Indirect Defense. The main objective of this study was to determine whether sticky plants that trap insects, as you can see in the image, can indirectly defend themselves by using the trapped insects or insect carrion as food for predatory arthropods. Specifically, the study tested if this carrion-based food source increases predator populations, reduces herbivory, and ultimately enhances plant reproductive success. Some of the research questions the authors focused on were, does adding insect carrion, dead Drosophila fruit flies, to sticky tarweed, Medea elegans plants, increase the abundance and diversity of predatory arthropods? The second research question being, does the presence of more predators on carrion-augmented plants lead to lower herbivore damage, particularly from the caterpillar Heliothodes diminutiva? And the third being, do plants that trap or receive added carrion show higher fitness, measured by the number of fruits produced compared to the control plants? The first hypothesis the authors came up with was entrapped insect carrion serves as a plant-provided food source that attracts more predators, especially species adapted to sticky surfaces, like assassin bugs, stilt bugs, and lynx spiders. The second hypothesis, increased predator activity from carrion addition leads to less herbivore damage by the caterpillar, Heliothodes diminutiva. And the third being, reduced herbivory increases plant lifetime fitness demonstrated by higher fruit production. The broader expectation of all of these hypotheses and the study being sticky plants benefit from a mutualistic interaction. Their glandular trichomes not only deter herbivores directly, but also support predators indirectly by offering carrion as a protein-rich resource. And then here I have a flow chart that I made of the hypothesized effects that this study would have based on the hypothesis of the authors. Some background information. Plants use both direct and indirect defenses to protect themselves from herbivores. While direct defenses like toxic chemicals or sticky trichomes physically deter or harm herbivores, indirect defenses rely on attracting predators and parasites that attack the herbivores. Common plant-provided foods that help attract these beneficial predators include pollen, extrafloral nectar, and protein bodies, which supply nutrients and encourage predator presence. The knowledge gap. Recent research has proposed that insect carrion, which is basically dead animals, but in this case, dead insects, could also serve as a valuable food source for predatory arthropods. But this idea had not been experimentally tested in a natural system before this study. Sticky plants that trap small insects with their glandular trichomes may unintentionally provide this carrion, creating a new kind of plant-provided resource that could strengthen indirect defense. This study investigated whether entrapped insect carrion on the sticky plant Medea elegans or common tarweed can enhance indirect plant defense by supporting predators that reduce herbivory. The researchers hypothesized that sticky plants benefit not only by directly deterring herbivores, but also by indirectly defending themselves by using the trapped insects as a protein-rich food source for predatory arthropods such as assassin bugs, stilt bugs, and spiders. And this image is from the actual article and study itself of the bugs in action, the predatory arthropods in actions, and some of the sticky trichomes on the tarweed. The importance of this study being that understanding this mechanism reveals a previously overlooked ecological interaction where plant stickiness may benefit both the plant and its predators, rather than simply just deterring insects. This work challenges the traditional idea that sticky trichomes are purely defensive and show how they can foster mutualistic relationships that improve plant survival and reproduction in nature. And here I have some images of what the sticky trichomes look like. Specifically, the image in the middle is the sticky trichomes on a Medea elegans tarweed plant. The study site and our organism. So this study was conducted at the Stebbins Cold Canyon Nature Reserve in Solano County, California. I have the county marked over here with the pinpoint on the map. It's basically an oak savanna and charpal kind of ecosystem. The researchers focused on Medea elegans or common tarweed. It's a native annual plant covered in glandular trichomes that make its surface sticky. The main herbivore studied was the caterpillar Heliothodes diminutiva, which feeds on the plant's buds and flowers, while common predators included assassin bugs, stilt bugs, and lynx and crab spiders, all capable of moving across sticky surfaces. So, for the experimental design, two complementary field experiments were conducted to test whether entrapped insect carrion enhances indirect plant defense. The first experiment being the carrion addition experiment, where the setup was 82 tarweed plants were randomly assigned to either control, which was no carrion added, or carrion added. The carrion being five freeze-killed Drosophila fruit flies placed weekly on the plant's trichomes. It lasted from early July through September, covering the plant's entire growing and reproductive season. And each week, the researchers recorded the number of predators, herbivore damage, which was the chewed buds, naturally trapped carrion, and fruit production as a measure of plant fitness. They also ranked each plant's stickiness or trichome density and secretion activity on a scale of 0 to 5. The second experiment being the caterpillar addition experiment, they had 14 pairs of additional plants were selected. One plant in each pair received a live Heliothodes diminutiva caterpillar, while the other served as a control without the caterpillar. To measure the direct effect of herbivory on plant fitness, that was the purpose. And then they had weekly observations of bud damage and fruit production to quantify how herbivory reduced reproductive success. The researchers condensed weekly data into season-long averages for each plant to evaluate treatment effects. They compared predator abundance, herbivore damage, and fruit yield between carrion added and control plants. Same with the caterpillar added and non-caterpillar plants. This design allowed them to isolate the effect of carrion availability on predator recruitment and indirect plant defense effectiveness. And this image right here is the flow chart from the actual article and study itself, where it shows the effects of the expected effects of their experimental designs and their data. So, trichome stickiness correlated with carrion and predator activity. Plants with denser glandular trichomes, stickier surfaces, entrapped more insect carrion overall. In control plants without the added carrion, those that naturally trapped more insects also had higher numbers of assassin bug eggs, showing that natural stickiness alone attracts predators and supports the reproduction. And then this image that we have over here is a diagram from the actual article and study. In the first diagram, we see how adding insect carrion, dead Drosophila flies, to the Medea elegans plants influence the ecosystem over the growing season. The top panel, panel A, shows that assassin bugs laid far more eggs on carrion-treated plants than on control plants, especially early in the season around June and July. This means that the presence of carrion quickly attracted predators and boosted their reproduction. In panel B, we see the opposite pattern with herbivory. As carrion and predators increased, the bud damage from the Heliothodes diminutiva caterpillars stayed much lower through most of the season. So this figure tells us that trapped insect carrion helped recruit beneficial predators early on, which then helped suppress herbivores later in the season. Predator abundance increased with carrion. Adding five dead Drosophila flies weekly to the Medea elegans plants resulted in a 76 to 450% increase in predator abundance compared to control plants. All five major predator species, the assassin bugs, the stilt bugs, the lynx spiders, and crab spiders, were found in greater numbers on carrion treated plants. This effect was strongest earlier in the season, which was June and July, when predators were colonizing new plants. And then here in this second diagram from the second diagram compares the control and carrion treated plants directly. In part A, you can see that all predator species, the assassin bugs, the stilt bugs, and spiders, were significantly more abundant on plants with added carrion. Some increases were dramatic, up to several times higher than on control plants. The stars on the bars indicate strong statistical significance, meaning these differences are real and not random. Then in part B, it shows the downstream effects. So carrion treated plants had fewer damaged buds and produced more fruits. This means that the added carrion not only boosted predator populations, but also led to less herbivory and higher reproductive success. Clear evidence of an indirect defense mechanism benefiting the plant. So reduce herbivory from specialist caterpillars. Carrion-enriched plants experienced a 60% decrease in bud damage caused by the Heliothodes diminutiva caterpillars, the dominant herbivore that feeds on these flower buds. This reduction in damage shows that the increase in predators effectively enhanced top-down control of herbivory. Conversely, when these caterpillars were experimentally added in a separate test, these plants suffered 50% greater bud damage and produced 50% fewer fruits, confirming that herbivory directly reduces plant reproductive success. And then higher fruit production and fitness. Plants with added carrion produced about 10% more dehisced fruits than control plants, indicating a measurable improvement in reproductive success or fitness because Medea elegans reproduces only through seed production. This fruit increase represents a significant gain in lifetime fitness. And then the third diagram from the study focuses on the caterpillar addition experiment. Here, the caterpillar was added to each experimental plant to test how herbivory alone could impact fitness. As shown in panel A, plants with a caterpillar suffered much higher bud damage, about 50% more than control plants. In panel B, those same plants produced half as many fruits. This result confirms how destructive herbivory is for Medea elegans plants, reinforcing that predator protection, like that provided by carrion attraction, is crucial for the plant's survival and reproduction. Together, these results show the full story. Carrion attracts predators, predators reduce herbivory, and plants ultimately produce more seeds and thrive better in the ecosystem. So, the study concludes that entrapment of insect carrion by sticky plants functions as an indirect plant defense mechanism. By trapping insects on their glandular trichomes, Medea elegans provides a supplemental food resource for a community of predatory arthropods, such as assassin bugs, stilt bugs, lynx spiders, and crab spiders. This carrion provisioning sufficiently increased predator abundance by 76 to 450% and reduced herbivore damage by 60%, leading to a 10% rise in fruit production and overall plant fitness. These findings demonstrate that sticky trichomes are not merely physical defenses, but also ecological tools that enhance top-down control of herbivores. The broader importance of these results and this study lies in showing that indirect defenses can arise from scavenging interactions rather than only mutualistic nectar or protein body systems. Because carrion provides a protein-rich resource without direct energetic cost to the plant, this mechanism helps explain why glandular and hooked trichomes are so widespread across flowering plants. It challenges the traditional view that sticky trichomes are costly due to predator interference and instead suggests that they may create an ecologically efficient trap system that benefits both plants and predator species. This discovery broadens our understanding of plant arthropod mutualisms and highlight how even accidental insect entrapment can contribute to ecosystem balance and evolutionary success. And then this is my reference where the study and paper comes from. Thank you so much for listening to my presentation.